Okay, so we're going to try to go through chapter 3 as quickly as possible. We're going to be talking about cells, their overall structure and function, and then we're going to get into some details about the organelles that are contained in the cell. So we kind of want to know what a cell's doing because that's an important part of how function takes place when we discuss organs and organ systems and things like that. Okay, so this term membrane can be confusing sometimes because it can actually refer to anything. Overall, it's a coating. It's something that's outside. So here you can see the pericardial membrane, which is actually made up of epithelial cells that coat the outer. It's an outer coating of the heart. We'll talk about that more when we cover cardiovascular. But what we're interested in right now is this thing, which is the cell membrane. And the cell membrane are made of these phospholipids. That's what these guys are. And then there are little cholesterol molecules, giving it some structure and support outside that. And then there are proteins sticking out, there are lipids sticking out, lots of stuff coming out to make up the cell membrane. So that's where we're going to start. Now, just as a review, we're all of the cells are bathed in some kind of fluid, and they have fluid inside them. The fluid inside, remember, is intracellular. The fluid on the outside is extracellular, so extracellular. But we sometimes call that interstitial fluid because that means that it's between the cells or around the cells. Now, in the blood, the fluid that we think of that's around, these are the blood cells, these little guys right here, and the fluid that's around that, we call that the plasma. Okay. All right. So if we start talking about membrane function, it does a lot of things. The cell membrane is hugely important, and it's going to keep coming back. That's why I talk about it. It's going to keep coming back up because we have to think about how things move. It's a bilayer, so these phospholipids, there are two of them with their tails together and their heads. That remember the hydrophilic head and the hydrophobic tail with the water out here. Okay, on contacting the head region. But a lot of things have to move through this, a lot of things are blocked by it, and so we have to kind of understand the cell membrane as a semi-permeable membrane. Okay, so some things can go in, some things cannot go in. It's permeable to it's selectively permeable. So it allows some particles to move through, but not all. Okay, so this allows things like a concentration gradient, which we're going to be talking about excessively from one side to the other. Concentration gradient, electrical gradient. So the outside of the cell tends to be positive while the inside tends to be more negative. And so all of these things. So the cell membrane, cells have to know where they are. They have to know where they are in relationship to each other. Sometimes or usually they're attached to one another. But a lot of communication, a lot of communication goes through exchange. It acts simply as a physical barrier to block things from moving through. Okay. All right. So if we look at if we really back out and we look at a cell membrane, it's very, very busy. Okay, so it's got a lot of stuff going on. So here we can see, if we look here, this is important. We can see these phospholipids and we call this a bilayer because here's a layer of phospholipids there, and there's another layer of phospholipids. So we call that a bilayer. It's two layers. And then there's fluid on the outside, like I said, and fluid on the inside. Okay, but see here we can see a glycoprotein. Glyco means sugar. Okay, so it's sugar. This is the sugar part here, and this is the protein part here. Okay, so it's a glycoprotein. And so we see other things. We see sugars that are anchored in by lipids. Okay, so these are fats. Remember the fatty acid tails that hang down from the head of a phospholipid? Well, here are fatty acid tails that are attached to sugars. Okay, so there's a lot of there are a lot of things going on. We would call that a glycolipid. Because of how it's structured. We have proteins that are transmembrane proteins that are going entirely through the membrane, the cell membrane. Okay, so these are transmembrane proteins. Okay, we have This is a lipid, it says here lipid anchored protein. So that's stuck there by these two little lipid tails. And there's a protein out there. This could be a signaling molecule. They just have lots and lots of different function. And then we see the cytoskeleton, which is attached to this protein, which is holding everything together. So you can see that all of these structures that make up the cell membrane are important for how it works. So this little picture over here is showing a protein. So this protein is that's an alpha helix, or it's moving through there, and then so you have part of the protein that's on the inside and part of the protein that's on the outside. This is great for communicating. So we can have a signal, we can activate something on this side, and it can actually cause something to happen on that side. Okay, so that's a way that the cell is communicating without letting anything in. So nothing is able to go in, but if a ligand were to bind this here, it could actually change its shape enough to make something happen here. Okay, and we call that a G protein. That's what this is an example of. There are lots of different types of proteins, but G proteins are the most popular in terms of drugs that affect them and things like that. Okay, so that's it. And here we see, this is a nice little picture of sphingolipids. Sphingolipids form little rafts down here that can sort of make a coating on the outside, which is further protection. Okay, so we have phospholipids that we just mentioned, sphingolipids, this is an example here, and then cholesterol, of course, which are these little guys here that sit in between the phospholipids, and they can be harvested to make things like steroids. Okay, so that's all part of the cell membrane. Okay, so but it's different for every cell, right? I mean, different cells are doing different things. So, for instance, here's the red blood cell membrane, which is about half and half proteins and lipids with some carbohydrates here. If you look at something we're going to talk about later called myelin, myelin is almost all lipids. Okay, so it's 79% lipids with just a few proteins. Inside the mitochondria, which we'll also talk about, it's a very busy place doing lots and lots of things. So it has a lot of proteins for that kinds of function. Okay, so for that function to take place, so it's very heavy on the protein, very light on the carbohydrate. Okay, sounds like a diet, doesn't it? Okay, so again, quick review. This is a phospholipid. We've seen this slide before. It has the polar head, which is hydrophilic. Trying to get that solidified in your brain. So polar head, hydrophilic, it likes the water. Okay. Nonpolar fatty acid tail does not like the water, and so will associate with another phospholipid that may be sitting down here, and then it's going to have down below, it's going to have its polar head. Okay, so that's what goes into making up. Primarily, the phospholipid goes into making up the membrane. Because if you were to dump these, if you were to make a bunch of these, which can be done, okay, if you were to make a bunch of these and you were just to dump them into um, a, a beaker of water, they would arrange naturally just like this, because the water is going to associate with these outer parts and it's going to push these tails all in together, and you're going to end up with something called a micelle. Now, if there's a pore, some of the water gets trapped in the middle of it, we call that a liposome. Okay, so it means body, so it's a lipid body. Okay, so and like I said, this little term here, it's a bilayer. Okay, so when you're wondering, and this is just how it is. I mean, these are just chemicals. You know, we've looked at the structure of fats, and then it has this little glycerol and then a little phosphate group, and that's what makes a phospholipid. Okay, so that's just it. That's the way they will naturally come together. You can dump this in a beaker, stir it up, and you'll see a bunch of these types of things that have formed. Okay, all right. So intracellular compartment, some terms here. The cytosol is the fluid. Okay, so if we draw a cell here, we can say that the cytosol is just the fluid part, which is going to be water. Then there are also inclusions, which are things like ribosomes, glycogen, which we've talked about. They all just kind of sit in here. And then there are organelles. Okay, organelles have their own little membrane-bound compartments in there. Okay, and so the all of these things together. So these are membrane-bound. I'm going to say that again. Membrane-bound. There's a nucleus. All of these things together make up the cytoplasm. Okay, so don't get, I know cytoplasm and cytosol sound very familiar. Keep them separate. Cytosol is the fluid. Cytoplasm is all of this, all of these things together. And then there's the nucleus that sits over here, and it's its own little structure doing its own little thing inside the cell. Okay, so yes, the cell is a busy place. So if we look at this, we think, first of all, we think, wow, that's kind of pretty. Uh, but then we can kind of, all cells, for the most part, we'll talk about some exceptions, but for the most part, every cell in your body, from neurons to your skin cells to liver cells, all of the cells in your body have these structures that are contained in them, and they all do specific things. This is a little factory. I mean, you've got your DNA that's hiding in the nucleus, and its job is to make proteins. Well, the endoplasmic reticulum is part of that protein making process. And then you can see here, this is the Golgi apparatus. Well, we have to excrete some proteins, sometimes out of the cell. So the cell will make the protein, and then it will release it, and it uses the Golgi apparatus to do that. We have these guys here, which are the mitochondria. And those are making the energy because this is very, very busy. So the mitochondria are busy making ATP, and then the ATP can be used by all the proteins and by everything else that's doing stuff. So all cells have most of these organelles at any one time. Now you can see your microvilli that are growing on the outside, and that increases some surface area, and we'll talk about that specifically as well. Not all cells have that, but all cells, with, like I said, the couple of exceptions that we'll talk about, all cells have these components in it. Now the cell might be shaped differently, but they're all going to have those things. So if we break these down, and we look at, and this is the difference here, organelles versus inclusions. Organelles have membranes, so the mitochondria, the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, these are just the digestive system of the cell. And the ER and the Golgi are used to make proteins, and then the mitochondria is used to make energy. These are the organelles. And then there are inclusions, okay? So it gives the example of lipid droplets, glycogen. Glycogen is that chain of glucose molecules that's used to store glucose, okay, for later use. And then ribosomes, and we'll talk about ribosomes are in the next section. Okay, and then there are a bunch of fibers, okay, so we can kind of see these here. We can see the little fibers that are giving some structure, and they're also something that they also serve other functions. These fibers that are in there. Centrioles are what you would call, to give it a fancy name, they're nucleation sites for mitochondria, but they're involved in or not mitochondria, but microtubules. Sorry. So they make microtubules, which are a type of cytoskeleton. Tubules. Okay, and so they produce these microtubules, cytoskeleton, and they serve other function too, but they're just long protein fibers. And then flagella, the only flagella that you'll find in the human body is on the sperm. Okay, so that's the only human cell that has a flagella, and the flagella is just a tail. It's the tail of the sperm. Okay, so this is what we're talking about. Cilia, to keep these separate, because a lot of people have trouble with this, cilia versus flagella. A cilia moves things, okay? So the cilia will move something, and it moves in such a way, for instance, on your respiratory system, the bronchi and the trachea have cilia, and these cilia are moving particles. So here's a particle that gets trapped in the mucus, and these cilia will move in this direction, and then kind of like this is trying to show. So then it's over here now, but then it kind of bends backwards and it sneaks back over to this side, moving this way, and then it moves it again. Okay, so it's moving something in one direction. So it's moving fluid in one direction. Okay, the flagella spins around, and this shows it snaking, okay, which is fine, because it does that, but it's to propel the cell forward. Okay, so it's actually propelling this cell forward by moving the fluid in that direction. Okay, so that's really the only difference. These things are both involved in movement. In the case of the flagella, it's in humans, it's moving the sperm, but this is also found in bacteria and other smaller organisms. Okay, and it's to propel. Okay, so it's to move it forward, and then the cilia is to move fluid. Okay, all right. Um, okay, so we just looked at cilia, and I'm spelling these out for you because it's confusing. Flagella, and now we have this other term, microvilli, because they all look kind of similar. I think we can kind of check flagella off. We could say, okay, flagella. I mean, that's the sperm, and this is the flagellum. Okay, we can get that. Cilia, remember, cilia is fluid movement and movement with my kindergarten handwriting there. Um, but now we have this thing called microvilli. Okay, now microvilli don't move. Okay, their job, and notice this because that's where you're going to get confused. I see it all the time. You're going to get confused between cilia and microvilli. Microvilli increase surface area. We're not saying anything about movement when we talk about microvilli. Okay, so that's where the trick question is going to come in. It's going to say, what's a microvilli? And one of the choices will be movement. And you're going to say, no, it's not that. That's cilia and flagella. Okay, but they have these microfilaments that kind of move up in there and give them structure. And then, like in the stomach, this gives a lot more surface area, so you can have absorption here as well as down here. Okay, so without these, we wouldn't have all of this surface area to absorb something. I mean, something that's flat like this can only absorb that much, but if you were to put microvilli, sorry, got to make sure I use the terms correctly. If you were to put microvilli, you can get a lot more surface area in that same distance, okay, between there and there. Okay, so that's its purpose. Okay, and microfilaments and microtubules are giving all of these things structure. So you can see these guys down here. They're the microtubules. There are the filaments. We'll talk about actin when we talk about muscles. And then there are intermediate filaments which sit down here. So these things are all, you can see now that the cell isn't just this empty little thing, it's actually got a lot going on. And these are just proteins, okay? So these are just proteins arranged in such a way that they're making these filaments and giving it structure. Okay, so one of the other things, I'm just going to say this briefly. There are certain things called motor proteins, which to me are just amazing little things. And this guy will walk along this fiber, okay? Right now, all you need to do is know that these things exist. However, when we're later on talking about how a muscle contracts, we're going to see that this is how your muscles contract. You have these motor proteins that are specialized to walk along a cell, or I'm sorry, walk along a fiber. In this case, in the case of muscle contractions, this is an actin fiber, and this is myosin. Okay, so we're going to learn that later on. But right now, if you just look at this, you can see this guy will keep changing its shape every time it binds, and it will walk along this filament in that direction. Okay, and of course it requires ATP, which is energy, to do it. Okay, so now let's start talking about some of the organelles. Speaking of ATP, ATP is energy. It's a molecule, and I'm not going to say too much about it here, but it's a molecule that has a lot of energy built up in its bonds. Okay, and so the body has figured out a way to use ATP as energy. Well, there's one place, well, primarily one place that makes great quantities of ATP, and that is the mitochondria. It's kind of a joke that the mitochondria is the powerhouse of the cell. Okay, well that's fine, but it's not very descriptive. So we're going to talk about how that energy is being made in a later chapter. But right now, you can see them. Here's a mitochondria. Here's a mitochondria. Here's a mitochondria. They have their own DNA, and you have your mother's mitochondria. Okay, so if you think about what happened at conception, the sperm moved into the egg, and all it did was release DNA. It released its own DNA. Well, there were mitochondria that were in here, and these mitochondria have their own DNA. Okay? And so that means as this thing started to divide, the only mitochondria that it had access to, because the sperm didn't give it any, the only mitochondria it had were these that were originally there. So you have your mom's mitochondria. Now, the idea is that the mitochondria, or mitochondrion, was originally just a bacteria, an organism that invaded other organisms, but it was so good at making ATP that other organisms started using it. Okay, and so now it is part of our cell. Okay, so it has its own DNA. It can't survive on its own. It's not a bacteria anymore, but it does have its own DNA, and this DNA is primarily used in some of these processes to make ATP. Okay, so that's the mitochondria, and that's what it does in general, and we're going to talk about how it does that later on. Okay, so it's arranged in such a way that it has this inner mitochondrial membrane, this inner membrane, and that's part of how this function takes place. So this is how ATP is generated. ATP is generated primarily down here in this area, and then it's released. Okay, so it has this unique structure, and that structure is that way so it can make ATP. All right, now we have the endoplasmic reticulum. Now, if we look at a cell, the endoplasmic reticulum is kind of shaped like this. Okay. All right. So the thing is, we have a couple of different kinds of ER. We have rough ER, which has a bunch of ribosomes sitting on it, and it looks rough. Okay. Now, I know we don't know what ribosomes are. Right now, it's just a word, or I'm assuming it is. Maybe you already know. But that's where proteins are made. Ribosomes are used, I call them translators. Okay, so these ribosomes are used to make proteins. So that's where, sorry, I'm going in the wrong order here, but the rough ER, protein assembly and modification. That's what it does. Okay. Now, something that happens over here, so this is rough, and it's rough because it has the ribosomes. And then over here we have smooth, no ribosomes. So if it's smooth and it doesn't have ribosomes, then it's not making proteins and protein assembly and modification. That's not its role. But if you think about a cell that grows and gets larger and larger, it needs more lipids. It needs more phospholipids. It needs fats. And so the smooth ER has assembly processes that allow it to make fats. It makes these fatty acids. sterols, lipids. So it makes all of these things. So that's what's going on with the smoothie R, and then those can move out here and they can become part of the membrane, or they can become cholesterol, or they can become, ultimately they can become things like hormones. Okay, so that's what's hormones. So that's what's going on in the smoothie R. So just keep those separated for right now. Smoothie R synthesis of fatty acids, steroids, lipids, modified forms in the liver, kidney, and the muscle. Um, but this is really the function that you need to keep separate, okay? Is the rough ER versus the smooth ER. And hopefully that'll make more sense when we start talking about protein synthesis. Okay, so here's another picture of this, and this is a nicer picture than I drew. These little blue dots are the ribosomes, and then over here you can see on the smooth ER it doesn't have the little dots because it is making fats, okay, specifically fatty acids. What's the difference? Well, fatty acids are specifically the tails, okay? Those are the little tails. So you can have something that's considered a fat, but specifically the fatty acid is this fatty acid tail that can then join to the glycerol to make, and what looks like what I drew here was a phospholipid. Okay, so that means it's going to have a little phosphate group on it too. All right, or it can be a triglyceride thing that looks like a jellyfish. Okay, all right, so that's the endoplasmic reticulum. Now, another thing. So if we look at this, if you can see this, okay, this little pink area here, that's the endoplasmic reticulum. This area right in here, that's the endoplasmic reticulum. So we're making proteins there. We just said that, making proteins or fats. But those proteins have to go somewhere. So they work with the Golgi complex or the Golgi apparatus. Maybe you've heard it termed that, which is this green thing right here. Okay, this is the Golgi. They also do protein modification, and we're going to talk about what protein modification means. It sounds like a pretty mysterious term right now. But they also do protein modification, but they do it so it's packaged into these little secretory vesicles. So if we look at a Golgi larger, what we see is that these proteins that are being made in here can all be packaged into this nice little secretory vesicle. And then if we have our lipid bilayer, our cell membrane here, then that can actually fuse with it and then release those proteins, okay? So when we talk about the Golgi, it's doing something specific, and it is modifying and then packaging proteins for secretion. Okay, so that's a major role of a Golgi complex. Because I might ask something like, well, gosh, what if you have a cell whose specialization is secreting things like mucus or something like that? Which organelle do you think is going to be very extensive in a cell like that? If you have a cell whose job it is to secrete things, it's going to have a very extensive, not just ER, but Golgi. It's going to have a very extensive Golgi complex, Golgi apparatus, because that's what it does, secretion. Okay, so this is another picture, a better picture, and it shows the same thing. It's in this case it's going up, but it's going to create these little secretory vesicles here, and then those are filled with proteins, and then they will fuse with the membrane. So if we have the membrane out here, it'll fuse with the membrane and then release those proteins to the outside, release those substances to the outside. Okay? All right. And then last but not least, the nucleus. The nucleus that's where your DNA is. Okay? That's where your own personal DNA is. Now, I did mention that the mitochondria has some DNA, But everything that makes you, aside from the few things that this mitochondrial DNA does, that's all contained in the nucleus. Okay, and they're arranged in chromosomes. Okay, so we have our 23 pairs of chromosomes that are all existing in the nucleus. Now, if we look at the nucleus, so I'm going to draw, this was supposed to just be the nucleus. I'm going to go ahead and erase that. But if we draw a cell and then we put the nucleus in it, what we see is not a bunch of nicely packed chromosomes individually. We see them all unwound and all over the place. Okay, so when this DNA, and there are a bunch of them, okay, but they're filling this up. And so when you look at it, you don't usually, unless the cell's about to divide, you don't see them as what we're typically used to seeing as chromosomes that look like this. Okay, that's a chromosome that's very dense and getting ready to divide. Usually they're unwound and they look like this, and so we call that chromatin. Okay. All right, and there's an envelope. We call this outer area the envelope of the nucleus, and it has holes in it. Okay, so it's got holes which are going to let the newly formed messenger RNA, we'll talk about what this is, let stuff move in and out. Okay. All right, so those are these three items: nuclear pores, the nuclear envelope which sits around it, and then the chromatin, which are the chromosomes that are all unwound and sitting in there. Okay, so we can see this, the nuclear envelope. This is a nice little picture. Nuclear envelope, the outside of it. Nucleolus, we haven't talked about that yet, but that's where there's a bunch of RNAs being made. And then this chromatin, it's DNA, and with the proteins on it that are transcribing that DNA. Okay, so that's what all of this is filled in here. That's the chromatin. And then of course these are the nuclear pores. Okay, the nuclear pores that are letting things out. Okay, that's what we see here. These arrows don't really line up very well. That arrow should be down a little further, but that's what it is. Okay. All right. Now, I'm not going to say too much about this, but it kind of puts everything together. And it says, okay, up here, we're going to go through all of these in a later chapter. So I'm going to have a section just on that. But right now, we're trying to get a big picture for what's going on. And we're saying, okay, here's the nucleus. That's where the DNA is. Well, the DNA never leaves the nucleus, but the purpose of DNA is to make proteins. But we just said if the purpose of DNA is to make proteins, it's not a protein, it's a nucleic acid. But if the purpose of it is to make proteins, but it exists inside the nucleus, remember what we just said? We just said that the ER is where protein synthesis takes place, and the Golgi apparatus modifies, the ER also modifies, but these things are modifying proteins. So it sounds like the protein work is being done out here, and that's absolutely true. So that means that the DNA, with its contained in the nucleus, makes a little copy. You know, for whatever protein you want to make, you just find a section of the DNA that has the code for that protein. You make a little copy of it, and that's RNA. And then these ribosomes will find that RNA and start making a protein. Now, this little purple thing right here, that's a protein being made. Okay? You can see it starting to be made on this one. Okay, so that's what the ribosomes are doing. So little by little, we're going to get into the details, like I said, and that's what all of this is. But little by little, we're kind of getting an idea. Okay, ribosomes are responsible, they play a role, that's what we know so far, in making proteins. That happens, it happens out here in the cytosol, but the ER, which is what we're just talking about as an organelle, they also will stick to this and they'll make proteins and they'll put them in there. And then these proteins, if we follow this protein kind of blebs off, that's a real word by the way, and then moves down to the Golgi where it's modified a little more, and then they're packaged into a secretory vesicle down here. So they move through this system, and then they're packaged into this little vesicle, and then those proteins are released. So we have an entire picture. We don't know all of the details yet, but we have an entire picture of how these guys are working together. Meanwhile, now, I will say that everything uses proteins. So we have proteins going to the mitochondria, we have them going to peroxisomes, which are involved in digestion and breaking down junk that builds up in the cell. Same with lysosomes. We have proteins that have to go to the mitochondria. So these are all being made and moved to different places. But here's the mitochondrion here that is producing what? It's making ATP. And because all of these processes that take place require ATP. Okay, so that's how they're all working together. We're making proteins. Proteins are the workers. I've said that lots of times. And they all need energy to be made and to function in a lot of cases. Okay, so that's what the mitochondrion is doing. So now we can kind of see how all of these things we just went over work together and they're actually doing something. Okay, so if we continue with some structure, there are some things to remember, and that is that when, let's say, if you're digesting something, you just ate a cheeseburger. This is supposed to be a cheeseburger, okay? And you break that down into little pieces. Those things have to be absorbed. If this is your intestine, those things have to be absorbed. We don't just want them to move between the cells. We don't want big gaps in the cell so these things just move through because we want to make sure that they're actually healthy for us. Okay, so we want to make sure that, like, for instance, if it's a sugar, that it's not a big complex carbohydrate. We want it to make sure it's a little monosaccharide. Okay, so it doesn't look like this. They are joined together by junctions. So most cells that exist in your body have junctions that are holding them together, and there are a couple of different kinds: gap junctions, tight junctions, and anchoring junctions. The one you have to know by name is this one, okay? The gap junction. I'm going to have a picture of that. Okay, so that's what this is showing. This is showing gap junctions, and so two things. First of all, it's a junction. Okay, this is a cell, and this is a cell. Okay, so these two cells are joined together. Okay, so that means it's a junction. That's what a junction does. It holds these two cells together. They're attached. However, a gap junction has a space where things can move from one side of the cell, from one cell to another cell. So they're a special type of junction. Okay, and they're a special type of junction that we are going to talk about, and you are going to be asked about on an exam when we talk about the heart. Okay, because gap junctions are what hold cardiac cells together, because communication is important in that case. And you see gap junctions in lots of other places, but the place we'll mainly be talking about it is in the heart. Okay, so it serves two purposes. It allows communication, but it's still a junction, so it's joining things together. Okay, so we can see more examples of this. Here's a tight junction. This is not allowing communication, and it is saying nope, nothing is getting through. So this is an example. Lumen of the intestine. The lumen is just the space of something that's hollow, like the intestine. And so this is the lumen of the intestine. So this is where your food is moving through, and you're trying to absorb it. Well, we want to make sure that it goes correctly and that it actually goes through the membrane, moves across the cell, and then is put in here. We don't want it to cheat and just start flowing through there. That would be bad because you could get a lot of toxic things or things that haven't been broken down properly, and that would be a bad thing. Okay, so we have these tight junctions that join them together. So you can see here, if you remember the gap junctions had this space, but if you look at this guy, this has no space. So nothing in this cell is going to move into this one. Okay, there's no way for it to move through. Okay, and there's no way for anything to come through in this way. It holds them together. Okay. All right, let's see, is that everything? Hopefully that gets that point across. Okay. All right, so to say a little bit more about this, because like I said, this stuff needs to be in your mind. because we have to start we have to start thinking about things as a process and as a machine almost, instead of just memorizing, terms and things to get us to the next section. We have to start thinking about these things in a functional way, um, because here you have what we sometimes call endothelial tissue, but it's a capillary, which is a very small blood vessel. Okay, so you have capillaries all over. That's why you blush. You have capillaries very close to the surface of your skin, but they're very thin and they absorb things. Things can move through the capillaries very easily. That's why you can breathe and things go into your bloodstream, because there's this nice little layer that allows things to move through. Okay, so we call that exchange epithelium. But here, with these exchange epithelia, you can see that this has an actual gap or a pore that allows some things to move through. Okay. Now these pores can actually close or open more to let more and more stuff flow through. Okay. So a lot of times when you hurt yourself and your ankle swells or something like that, what's happened is that this guy has shrunk a little bit and it's letting more fluid flow into that area. Okay, and that's causing a fluid buildup and you have swelling, edema. Okay, so that's what that is. All right, so we're going to see this. We need to think about it in that way. We need to think, okay, well, there can be tight junctions that do not let anything through, and then we can have this exchange where in some cases we can actually have flow through here. Okay, we don't want that in the intestines. We do want it sometimes in capillaries. We want to allow that fluid to move through there. So there are modifications and adjustments that can be made for that. Okay. Um, do you remember the difference between cilia and microvilli? It's a little quiz there. Cilia is fluid movement. So hopefully you got it. Microvilli are for surface area. Okay, so in this case, this cell has both. Okay, ciliated epithelium lining the airways. Okay, so it has both microvilli and the cilia. Okay, now it also has these items here. There's the Golgi. There would be the endoplasmic reticulum. So it's still a cell. It still has all of these things. This could be secreting mucus. Usually you have specialized cells for secreting mucus. Um, but that's an example of a cell function. Okay, and then of course you have the mitochondria because everything needs ATP. All right. Okay, secretory epithelia. I talk about this because one thing that we do is we do things like we sweat and we excrete fluids. We excrete fluids from mucus in our GI system. Actually, in most places, you're excreting mucus. You have lots of things that are being excreted, and it's kind of an interesting idea if you don't actually talk about what it is that's happening. Um, by that I mean, if you were to ask somebody, how do things get excreted? Okay, how do sweat glands work? They might think, oh, well, there's fluid inside it that builds up and then there's a muscle that squeezes it out. Well, that's not the way it happens. That's not the way secretion happens. The way secretion happens is these cellular processes will produce something and it fills this little pore, and more and more and more of it builds up and it starts to ooze out. Okay, so that's the way secretion takes place. Okay, and so a lot of times there will be specialized cells called goblet cells that will sit in here, and they're the main ones that make these fluids. And then it oozes out. Endocrine, we haven't talked about endocrine versus exocrine yet, but sometimes we're releasing things into the blood, and so it's kind of the same thing. We just build these things and then we release them, usually through interstitial fluid, and then they move into this capillary and it oozes into the blood. Okay, so that's secretion. Okay, so that's secretion. When something is moving into the blood, we say that it's being secreted. All right. And then the last thing we're going to talk about is apoptosis, okay, cell death and replacement. If you have, if you know someone or if you have a patient or something that has a stroke, a lot of times you have cells that die, okay? So if a neuron dies, like because it doesn't have enough oxygen, okay? So if a neuron dies, we'll say this is a neuron, what it necrosis means that it dies and it releases all of the goo and all of the proteins and all of these enzymes that were inside it. It releases those, and those are toxic. Okay, that's supposed to be a skull and crossbones. Those things tend to be toxic. That's very, very bad. Well, the thing is, cells get old all the time. Okay, they function for a little while and then they stop functioning, and whether it's through DNA mutations or whatever, buildup of toxins inside them, they stop functioning and they don't work anymore. They don't just die and leak out all of their stuff, all of their toxic stuff. That would be bad. What they do is they go through a process called apoptosis. This guy gets a signal, says, hey dude, you're too old, you're not working right anymore, and then it will just sort of fall apart. It'll start to separate and it'll turn into particles and it'll contain all that stuff. And then that can be broken up by peroxisomes or macrophages. Something will come in and then detoxify all of that stuff, turn it into something harmless, and then release it. Okay, so that's what apoptosis is. Apoptosis is the term for programmed cell death, cell suicide. It does it in such a way where it doesn't kill all of the cells around it. Because if you have a neuron that dies in that way, it could potentially kill other neurons. And that's actually a big problem after a stroke, is that the stroke may kill a certain number of neurons, but those neurons, upon dying, kill other neurons. So you have to stop that. You have to stop that process, and that's one of the things with stroke that they're working on, is how do we, limit the damage to just the cells that were initially killed. Okay. Replacement of cells, stem cells. We're going to talk about this again later too. Stem cells they retain their ability to divide. So they're sitting down here, and so when these cells die, so this cell goes away, it's gone, then we can have this one divide. Okay, so it'll divide, and then this one will go on to take its place. Okay, so this can happen in some tissues. It happens indefinitely a lot, but there are limitations sometimes to how many times this can divide before it's done. Okay, all right. And then research uses and potential. We've heard a lot about stem cell studies where if you put stem cells in, they can maybe replace lost neurons. Because right now, once these guys are gone, they're not really replaced, at least not in the central nervous system. Okay, so that's all we have for cells. Hopefully that wasn't. I know it's long. I'm sorry about that, but hopefully it's helpful. Okay.